Hi, I'm Brad Power, and you've arrived at the Linnea Olson Hackathon. Today's agenda is the last week was the kickoff, but this is the kickoff of the medical records, the platform, the foundation for the journey that we're going to go on in this hackathon. And Linnea is going to introduce us to her medical history. She's going to talk about what data she has that she can make available for analysis, questions that she has, issues, and then so that's the first section. That should take about 15 minutes. And then we're going to take about 15 minutes for questions of clarification from the audience, suggestions about additional data that might be useful, and just sort of, kind of process what we've heard and where we go from here. And hopefully we'll finish that in about half an hour, which is longer than our usual lightning updates. And then we're going to have another half hour for those that want to, an optional addition for people to introduce themselves and get to know each other. We had that last time, and it was really inspiring and powerful to hear the diversity of people that are participating in this hackathon. And just like you go to a conference and you meet some people, kind of some peer-to-peer networking, we're going to add that at the end as an additional feature. So with that, Linnea, why don't you walk us through the medical story?
Sure. Hello, everyone. My first update is that on Wednesday, I had blood draws for 2 blood biopsies with both Foundation Medicine and Lucent. So hopefully by next week, there will be some more information as far as genetic profiling. So that's super exciting. When Brad made reference to my records, I have all my radiology CDs. I have 5 years worth of records, but I'm going to request the other 10 because it looks as if we now have a place potentially to store it, right, Brad?
Yes, we actually have Kim Rees on this call and Tim Stollmiller is on this call. So we have a couple of candidates for folks someone here from Citizen. So we have Citizen, XCures Cancer Commons, and Path Presenter all as potential places to host your data, and in particular all those images, those many images that you have. So we're going to actually have a conversation later today with Kimmery, and I don't know if Tim's going to join us, but I think Marty Tenenbaum. So we're going to figure that out. Figure out what's the best solution, but we're going to certainly have something because we have a number of folks that have come forward to help.
All right, great. Well, here's the verbal description of my journey, I guess you could call it. And Brad and I have talked about the fact that hopefully by next week, I will have this presented in a graphic format so that you can actually see it. But today I'm going to give you an overview. So I was diagnosed with non-small cell lung cancer on 4/5/05. 2 weeks later, I had a lower left lobectomy, and my path report post-biopsy was bronchio, I can never say this, low alveolar variant mucinous, which is now referred to as invasive mucinous adenocarcinoma. So my tumor was 5 centimeters with microscopic airway spread. I had clear margins, lymphatic invasion not present, lymphatic infiltration in tumor was slight. 21 lymph nodes were removed, none contained tumor, and I was T2, NO, and they did do EGFR sequencing, which was pretty phenomenal back in 2005, but I was negative for that. So in June, about 6 weeks after my surgery, I had 4 rounds of cisplatin and taxotere. And then my first scan post chemo was in September, and I already had a new nodule on my left lung. So progression, but we didn't characterize it as such yet. And we entered a period of watch and wait. 3 years later, I was very symptomatic again. And in June of 2008, I was biopsied and restaged to 4, also told I had 3 to 5 months left to live. Just as sort of a last ditch effort, I went on Tarceva, and my oncologist later explained that this was in case the testing had been mistaken. In August, 2 months later, I was informed that the biopsy I had been there had been tissue submitted for genetic testing, and I had come back positive for EML4 ALK translocation. And it was explained to me at this time that there was a phase 1 clinical trial available. So on October 1st of 2008, I went on crizotinib. I had a 70% response, which was phenomenal. Progression after 1 year, but I stayed on for 2 more years because there was no other treatment. At 3 years, I was biopsied again and found to have an S1206Y secondary acquired mutation. At this point, I started my second phase 1 trial in October of 2011 for ceritinib. Again, I had a 70% response. While on ceritinib, I did have liver toxicity at one point. We were able to get me back on drug, but at 15 months, I had progression and had to come off. Biopsied again. This time S1206Y was not found, but in its place was G1202R, which was bad news because it was resistant to almost all ALK inhibitors. I was also tested for PD-L1, but negative. So in December of 2012, I started on taxotere and pemetrexed, 6 rounds of that, and then just pemetrexed for 6 months. And then I asked for a break. So now we're in 2014, and I go back on crizotinib for 2 months in order to qualify for the next trial I will be joining. And I joined that trial, another phase 1 trial, in May of 2014, and it's for lorlatinib. Once again, I have a 70% response. And then I have this wonderful holiday time where, frankly, I almost felt like I was cured because I had stability for 4 years, and it was just astounding. I will add, though, that in December of 2016, my MRI showed that I had gadolinium present in my brain. And I just add that as an incidental finding. So in November of 2018, I had progression yet again. And a biopsy showed that I now had, in addition to G1202R, G1269A, and S1206F as mutations. So I stayed on lorlatinib for one more year. And then in December of 2019, we added carboplatin and pemetrexed to the lorlatinib. But I had progression anyway. And in February of 2020, I started another phase 1 trial for DS1062A. And this is a trophoblast cell surface antigen 2, TROP2, targeting antibody drug conjugate. I had some stability on this, but eventually progression. And so in November of the same year, I started yet another phase 1 trial for lorlatinib plus binimetinib, a MEK inhibitor. And this time I had a 57% response. However, after a few months, it was shown that I had ocular toxicity, blisters on my retinas, and I was kicked out of the trial. And now I am just on lorlatinib. So as far as what I have talked to my oncologist about for the future, it looks like there's potential for a SHP2 inhibitor plus lorlatinib. I am concerned about this because it's the same pathway as the MEK inhibitor. So once again, there's the potential for ocular toxicity. Another drug that I have been interested in is Turning Point's TPX0131. It's a next generation ALK inhibitor. It's a macrocyclic structure that can bind completely with an ATP binding. It just opened phase 1 in Australia, but the problem is that patients who have been failed by more than 2 TKIs are not eligible. So I would not be eligible for that. And it looks like phase 2 won't start for 18 to 24 months. So right now, that's where I'm at. I'm on lorlatinib and trying to figure out what the next treatment will be.
Okay, so this is where comments, questions, I'm hoping someone like Kimmery in particular, who has some knowledge of lung cancer, might, summarize or comment or question. And if anybody has ideas, for things, additional data that would help, or hypotheses or all that, this is the time to kind of process and digest what Linnea just laid out. Kimmery, do you have anything?
Yeah, I want to start just with a question. So Linnea, technically right now, are you considered to be in stable disease or progressive?
No, right now well, actually, you know what? I guess I can't answer that question, but I think the assumption is that I probably will be progressing now that I'm just on lorlatinib, since in the past I was resistant to this. Mm-hmm. And I stayed on it only with the hope that maybe some of my cancer is still responsive to it.
Okay. And I have a question about the MEK inhibitor trial. Did you explore the possibility of compassionate use of a different dosing schedule of the MEK inhibitor? And the same question for the TPX0131. Did you inquire about compassionate use of that drug?
No, but that is a possibility. As far as the MEK inhibitor, I think that when I spoke to the retinal specialist, she said, because I asked about intermittent dosing, and she said that were I to go on it again at all, there was the potential for more damage. And of course, ultimately it could have led to blindness.
Yeah.
So this was something I took seriously.
And was the toxicity, I mean, when you discontinued, did it reverse completely, or do you still have some lasting effects?
I have a little bit left, but it is reversing, and they have done a good job of checking up on my eyes. So, and it's interesting, I should add, in that trial, as far as I know, I'm the only person who had response. Everybody else, the best result was stability. I'm also the only person who had ocular toxicity.
So, yeah, I mean, that speaks to me that the dosing they need to work on the dosing a little bit more.
Yeah.
But with no response.
And
Any other questions?
Just a question, is the MEK inhibitor an experimental MEK inhibitor or one that's already on the market?
It's something that is used in melanoma.
Aha. So,
But yeah, it's a new thing to try it in lung cancer.
yeah.
Believe it's
Sorry.
approved in combination, I think, with a BRAF inhibitor.
Yeah, okay, that one. I mean, technically it could be used off-label in a experimental way by a brave oncologist.
So who's your oncologist? Where are you being treated with all of this? Has it all been the same person?
I've had 3 oncologists, all extraordinary. I'm at Massachusetts General Hospital, and I had Dr. Tom Lynch initially, then Dr. Alice Shaw, and Alice still weighs in on my case, but she's mostly at Novartis now. And now I have Dr. Jessica Lynn.
Grace Cordovano has her hand up. She's and others, if you want, can use the raise hand feature. Grace.
Well, first I wanted to say, I think you're absolutely amazing in how you present all of the intricate details and nuances. So I'm blown away listening to you. Not that anyone is surprised, but I just wanted to recognize that when I hear doctors say, well, patients don't know these things and they don't need to know all these things and they just have to trust us. So there's that. But I wanted to ask, and I apologize if I missed it in your details, when was the last time you had a biopsy that was sequenced?
So I would have had a biopsy, 2 biopsies actually a year ago as a requirement. And I should have put this in my presentation, but it was a requirement for, oh geez, which trial? The DS-1062A.
Okay.
And unfortunately, those biopsies were research biopsies rather than clinical.
Mhm.
And after all these years of being a patient, that's the first time I learned that when it was a research biopsy, I would not be getting any return on the information. So my
Is there
tissue.
any way we can appeal that because of COVID?
I appealed it the second time. Once I learned, I just told the surgeon it was bullshit and asked her to see if she could get some additional tissue for MGH. She did. However, my path report on that simply says I have cancer. So
What
not useful.
do you withdraw your consent on that research? You could formally withdraw your consent for further use of any of your remaining tissue for research.
Really? Okay. Now, and this was not part of the presentation either, but I should add that in the past, I have been non-compliant in my trials for various reasons, including excessive scanning. And then I was concerned about the gadolinium in the contrast, and rightfully so, as I eventually was positive for it.
Linnea, so my question as a follow-up to that is, do you have any thoughts or wishes on having a more recent biopsy done for some type of sequencing? I would defer to the experts in the audience, whether it's NGS, whether it's foundational one, whatever it is, to see if there's been anything that's been activated that could be a consideration.
If it were optional, I would have one. And this is something maybe I need to discuss further with my oncologist, but to me, it's incredibly irritating and insulting that the most recent biopsies I had didn't benefit me in any way whatsoever. So I wish there was a way to just opt in and say, I want to do this, but I want to do it so that the information helps me.
Mm-hmm.
Is
Yeah.
it possible that you could request the actual tissue that was taken, used in that biopsy, so that it could be sequenced as might be valuable? And, with obviously the proviso that you get that information.
Well, it went to Japan, and my guess is that there probably was no excess because these are needle core biopsies. So, it's tiny little biopsy anyway.
Yeah.
But yeah, if there's a way to get a biopsy now so that it could be used for this hackathon, I'd be all in.
Tim, if you wouldn't mind, you've been putting some great information in the chat. Would you just kind of verbalize it for all of us? Thanks.
Yeah, absolutely. I mean, there's a number of different options in the MAPK pathway. The fact that you got some ocular toxicity means it might be an actual pathway issue, because MEK inhibitors do tend to have some eye tox, and so not necessarily the best option. But I do just want to raise awareness that there's a number of approved MEK inhibitors. I'm not 100% sure of their precise safety profile. Ilixertinib is the first-in-class ERK inhibitor, the next down in the pathway. And XCure is actually running the expanded access program for Biomed Valley. And this is a pharma company that's sort of a subsidiary of a large nonprofit research institute. And so they're very open in sort of their treatment modalities, methodologies. And so the program that we're running is actually extremely open-ended. It's essentially any patient with a MAPK pathway activating alteration with a solid tumor. And so essentially it's a basket trial for MAPK pathway alterations. And on top of that, the protocol is written really open so that the treating oncologist can use combinations with approved agents, either on target, on label, or even off label. And so really it's a sort of essentially an open label basket study, but it's a mechanism to access ulixertinib. So if a MAPK pathway targeting option is good for you, this is a way to combine it along with other approved drugs like lorlatinib, for example. Again, I'm not saying that that's going to be a great option because you had talks to that in the past, but
right.
I just want to point that. And, compassionate access and expanded use, I mean, that's an excellent way to explore novel combinations if the company is interested in making their drug available. So, and I will say, there's a number of trials with other ERK inhibitors. I think maybe I can't remember which company, but they're also doing SHP inhibitor combinations, SHP2 combinations with MEK inhibitors. So,
something else that I wish could be explored, but there does not seem to be a lot of leeway on is dosing, where I wish pulsing was an option, because as far out as I am on the branch, it seems that combination therapies are now the suggested route to go. And I have to tell you, the toxicity is difficult. You know, I mentioned the retinal blisters, but I had so many issues with binimetinib and lorlatinib. And it was interesting. They were surprised because neither of the drugs on its own is so toxic, but combined, it just exacerbated the situation.
Yeah, that's why I was wondering if you could, find an oncologist who might be willing to, if you're willing to risk those even minor toxicities again, they're just to be able to do more metronomic kind of sequencing or dosing.
Right.
That's completely experimental, obviously.
Right.
But I mean, I agree with you. I mean, having worked at pharma companies for a long time, that was one thing that I always worried about too, is, the dosing studies are done for particular purpose to get, a single agent approved usually, and then you use them in combination with each other at their approved dosages. And there's not a lot of, I guess, there's not enough, I think that's my criticism. There's not enough work done on, dosing, combination dosing, right?
Right. Yeah, and I think there does need to be more, because obviously more and more people are going to be taking these combinations, but the side effects I had from binimetinib and lorlatinib, I told my oncologist, this is not tolerable for most people.
Emily, you had a comment in the chat. Can you speak to that ALK positive research in this area? Yeah.
I'm part of the ALK positive patient group. And I can't remember, Linnea, were you on our first call with Turning Point?
I was not.
Okay, so we have been talking to Turning Point, who is working on this 4th generation ALK inhibitor, TPX-0131. And that was one of the first things we asked them was, will they have a compassionate access program? Because there are so many patients like Linnea who have been on more than 2 TKIs and now need this drug as kind of their next best option. And they said there are a lot of, I don't know much about the FDA regulations for compassionate use. And they said they at least have to find their dose first. So they're starting the trial in Australia with the dose finding phase of the phase 1 trial. So I hope sooner rather than later they'll have that, but it's not clear when that would be. And I don't know what the pathway would be to speeding along compassionate use.
Emily,
So
Colin had said something about the cohorts just having one person. Is that true, rather than 3?
That's in the early stages. Yes, they're starting with a very low dose and then moving up. And at some point they'll expand it to 3 per cohort.
And Emily, this is Willie with XCare's work with Tim.
Sure.
I think one thing I can add, just in case it enters into that conversation, is that the waiting until they have a dose, there's probably some nuance to that if they're at a dose where they have some safety information
Mm-hmm.
and they're still escalating, but they also believe they're at a dose that could have some efficacy, that in theory is sufficient to justify expanded access in parallel with continuing. It takes risk tolerance by the company and everything to start more things in parallel. But in this disease with the ALK history and such, and the potential to move faster, that's just a nuance in there that people may not realize is possible if they're truly trying to find a solution. There may be an opportunity to move faster in parallel.
Okay. Good.
I'm going to stop the recording now because we've a number of people have had to drop off. We will continue talking, but I'm going to stop the recording, and we can, like, there's still room for more questions if you like.